while, and instead, sage can mean several things. It could be a profoundly wise person, judicious or prudent, like Albert Einstein perhaps is a very good example of a sage. A bioinformatics sage could be someone like this guy sitting on top of the mountain with his solar panels and arrays and satellite dishes. And he says, then, once you discover what it is you are searching for, just type it into the search engine. Well, today we're going to use this definition of SAGE, which is serial analysis of gene expression. Now, here's the basic concept. You have a biological issue. Let's say you start with a single cell, and then it divides by mitosis to give two daughter cells. Now, those two daughter cells contain identical genomic DNA. But in a variety of situations, those two cells can start to differentiate from each other. During development of an embryo, of course, that's one logical place where one cell would grow and differentiate finally into a neuron, and the other cell could, depending upon the signals it received, differentiate into an epithelial cell. Another important thing that this is being used for is when cells start to divide. If one of the cells becomes mutated and turns into a cancer, then you would have two cells with virtually identical DNA, but one of them is now dividing out of control. So the analysis of differential gene expression, or SAGE, can let you try to understand what is changing or what is different between these two cells. If the genomic DNA is the same, something has to be different about these cells, and that will show up in the expression of the RNAs and the proteins that are made. So bioinformatics is now trying to build tools so that this really complex topic can be understood better. Historically, this kind of thing was studied by scientists one gene at a time, and I think the overwhelming majority of the time it's still studied in that way, where you study one gene like SARC or RAS or a kinase. However, now you can start studying multiple genes at a time. So we're going to talk about that, and that's what today essentially is about, is how to study these multiple large sets of genes. This little tiny figure on the left in red and black is showing the expression of 216 different genes analyzed by SAGE. The different levels of expression are shown in the intensity of the red color. And so even where it's all black, there are these tiny little squares, and each square is filled with the cDNA from a particular gene. So the bright red, pinkish color are the genes that are being expressed at the highest level, and those in black are being expressed at the lowest level, or not at all. This is how you would set up some sort of microarray, particularly if you're doing SAGE and comparing two different tissues together. In this particular example, they're showing us how you could compare gene expression from a normal sample to that from a tumor. The initial first step is to extract the messenger RNA from the cell, and then you use reverse transcription to make it into a DNA. You can do this by PCR, and when you're amplifying the cDNA, you incorporate some sort of fluorescent label. And here in this example, the normal cells have been labeled with a green fluorescent dye, so it fluoresces in the green wavelength, and then the tumor tissue has been labeled with a red fluorescent dye. You take the two samples now that have been copied back into DNA, and you mix them together in equal ratios as much as possible. Then you incubate that with a microarray. So on the right hand side of the figure, that shows you the microarray that is set up with a robot. And so it takes samples of bacteria or samples of DNA from a plate, it transfers it to some sort of solid support. In this case, they're showing us what looks like a microscope slide, and the robot dots it in a particular pattern so that you can find and know which position each DNA has been placed. Then you hybridize your probe to the array so that it it will find complementary DNA sequences and bind to it. Then you rinse it really good, and then you scan it on a fluorescent reader, and you get that little tiny square shown on the bottom right, where if you see red, that means the tumor is expressing that RNA, but the normal cells are not. If you see green, that means the normal cells are making that RNA, but the tumor cells are not. And if you see yellow, both cells are expressing it. And of course, you can get different ratios of color. Here's a real example. So in this case, there are 16 different plates arrayed together on the same array. There's approximately 10,000 different spots on this panel, and so some of them are controls and negative and positive controls, but the rest are known genes from a sample, and then it's been screened with the probes like we talked about in the last slide. And so you can see, if you look closely, examples where there's green by itself and red by itself, and there's yellow and orange and all different levels in between. So you would read this with a scanner, and it would give you a report about the different levels of expression based upon the wavelength of light that was observed. This is a pretty powerful technique. It can answer quite a few questions. For example, it can tell how many and which genes are expressed in different tissues. It can tell what genes are expressed at a high level in different tissue types. Um, this is also going to give us some idea to be able to define what is meant by a high level. Is that 10 RNA transcripts per cell, 100, 10,000? And so people are trying to answer that question. That's quite a difficult question to truly answer. You can also try to understand what fraction of total genes are expressed in given cells. How does gene expression change as cells progress through the cell cycle? How does gene expression change in diseases? For example, we just talked about cancer or brain tumor. You can talk about other disease states as well. How does the cell gene expression change during infection? How does gene expression change in a variety of situations? So it's super useful. How is it done? We're about to go and look at the databases, and these databases that are being built up are taken from scientific studies done by labs all around the world. And the methodology for SAGE is, it doesn't, to generate this, instead of using a probe that covers the entire gene, they take what's called a short sequence tag. So from the entire gene, you look within the gene for regions that are completely unique to that gene. Often 10 to 14 base pairs is sufficient to make it unique and different from all the other RNAs. So you find those unique tags. And in many cases, you can take those tags and link them together to form long molecules that can be cloned and sequenced and then used to generate these probes. But by using these tags, then instead of the entire RNA, you can have a better idea of specificity, so that you're only binding to your RNA of interest or your DNA of interest, and you're not getting cross binding to other genes. There's two papers I'm showing here in this particular slide. You can go look them up if you want to learn more about this idea. Then this data is being placed in a couple different areas, but it's all brought together in this GEO database, which is at the National Center for Biotechnology Information. So we're going to be back at NCBI and the NIH. This GEO stands for the Gene Expression Omnibus, and people upload their data so that it can be accessed by the public using different search tools to display it and understand the data in a variety of different ways. You can do lots of things. I'm only going to be able to show you a few different pretty cool things that you can do. This is the paper here detailing the development of the GEO database and some of the tools. This is from 2006, so it's certainly been updated a bit and changed some by now, but this gives you at least the link to the initial first paper. Now, you can access the GEO database in a variety of different ways. In this case, I've gone to NCBI and gone to the GEO database page. You can link to that from the main page. You can also go to the GEO database like we've done everything else by going to NCBI and doing an all database search, and that will get you linked to the GEO database. But here, let's start out on the page, the main page for GEO. There are a variety of different things that you can do from this main page. Our interest in this class will be searching for stuff. But if you look at this, you can look down at the bottom. There's also a submitter place. So if you're someone who does gene expression experiments, you can upload your data here, and you'll have an account and be able to do things. The formatting and uploading of the different types of data is complicated, so they have a very strict set of rules for doing that. But we aren't going to be concerned with that. We want to search the data set. So the first thing I'm going to search is under the query section here. I'm going to search the data sets for those that are concerned with humans and smoking. And let's see if there's anything in the database with that. So you type in your keywords here, click on Go. Now I found, in this case, 94 records. Now I did this search, actually, I'm giving this lecture to you from a PowerPoint slide, not from the live database. And so I'll be interested, if you guys want to, you can do this same search. Now, this has exploded in the last year, and my guess is that if you do the same search now, you're going to get over 1,000 records. Be that as it may, the information is the same from earlier this year as it is now. You get a bunch of different records. So it's organized into records, and they all have a unique number, which is in this case, this one is GDS 2771. That stands for the geo data set. It's a particular data set. This one is an analysis of large airway epithelial cells from cigarette smokers with and without cancer. And so they want to compare these two. Inside of this data set, we're going to look at it in just a second, is information taken from a single study. This one gives you a little summary. In this case, there are 192 different samples. What that means is there's 192 different people from which they've taken samples, extracted RNA, and then screened it using these different sequence tags looking for specific genes. So if I click on that record, it takes us to the data set browser page, and you can look at a bunch of different things about the data set. We're going to go through them one by one. Initially I'm going to click on the far right where it says cluster analysis. So let's look at that. This shows us the data that appeared and came sort of raw data from the paper itself. They've labeled different genes, as we mentioned before, from normal tissue, and also from different individual samples. And some of them are red, some of them were labeled green, and then they screened their arrays. Let's zoom in on a little bit so we can get some detail here. This doesn't make any sense to us right now. I'm just showing it to you to give you an idea of the massive scale. Along the top, you see all those individual lines. Those are this 192 data sets. That's on the horizontal axis. On the vertical axis, those are individual genes that are organized in gene families together. So there's so many that it just looks like a big black mess. But if we zoom in, we can start to see some detail. So again, I've just taken a screenshot of a small area here along the top. Those are individual records, so from individual people. And from this particular data set, and you can look on the right hand side in the column format are individual genes from the human genome, and then the level of expression from the individual sample in red or green or some combination thereof, or black. And so these data sets are massive. What these are being used for is to search for genes and patterns of gene expression that could either be used as a diagnostic tool to determine if a cell is becoming cancerous or not, or to look for individual genes that might be as a treatment option. So let's say one particular type of cancer associated with smoking always has one gene that's turned on at a really high level, and then that would be a natural target to go after. Okay, let's back up now, back to the dataset browser. Let's say I'm interested in a single gene. See what I've circled down there towards the middle? I want to look at RAS, because RAS is involved in almost every cancer. So let's see if it's doing anything in this data set. So you can type your gene name in here. whatever you're interested in, and click on Go. Now we've searched this record, and so these are individual records within our initial data set. Okay, these are the one, remember there was 192 samples. You can see that right here. These now are showing individual genes found within these samples. Now if I click on the figure, it's expanded that figure. Now this looks at the level of expression from the individual persons in this database record, and you can see that the expression is all over the place for RAS. There's no clear pattern. On the left, those with the line underneath where it says right here, these are those that do not have cancer. These are individual people that do have cancer, and what they're looking for are patterns. That's not apparent in this particular sample, but sometimes you'll see really high levels of gene expression here, and then in the alternate, really low levels over there. That's what they're looking for. Well, let's come back out now. I clicked on one of those. This is an individual record from that data set, and this was a smoker diagnosed with cancer. They took bronchial epithelial cells, extracted RNA using Trizol. They tell you how you do it, and then they screened looking for gene expression at these different levels. It tells you when the data was submitted. It tells you who did it. So this was a scientist named Avram Spira. Gives you his or her email address and phone number and everything. So what they're trying to do here is develop a community of people that are interacting with each other, sharing data to try to learn more about treating cancer or treating any kind of disease. Back here, here's another sample from the same data set just to show you the difference. This is a person that was not diagnosed with cancer. So there's a lot of information about every single record from which the RNA was taken that you could use when you screen these samples. Now these data sets are still on there and they can still be searched. So there's all kinds of information hidden within these records that somebody needs to find the right question to ask, and you can still continually use this data over and over again to try to learn new things. Now let's come back out to, that's the GEO data set. It is associated with this database here called CGAP, which stands for the Cancer Genome Anatomy Project. The GEO data set is all types of data. It can be involved with developmental biology, it could be involved with cystic fibrosis, it could be involved with just normal tissue in response to environmental changes. But those that are concerned just with cancer have been segregated or at least placed in this very certain special database that is focused just on cancer. This data is available to everybody around the world, anybody interested in cancer, and screening all of this data. And they developed these tools to allow us to do lots of things. So shown over here on the left, we're going to spend all of our time really using this SAGE Genie. Okay, so we're back to serial analysis of gene expression, and the SAGE Genie will allow us to search all this data in a very convenient way. So I clicked on the link, and here we are at the SAGE Genie. There's lots of different tools. As you can scroll up and down, and I really strongly encourage you to play with this and look at all the different things. There's a bunch of different things that you can do. A lot of it is hard to use and complicated, so I'm going to stick to some of the major features. But you have to become an expert to be able to really use this database to its full levels. The one we're going to use a lot is the SAGE Anatomic Viewer. So click on that link right there. Oops. Here we go. This will take you to another page. I've zoomed in just to show you one particular area. You can search the Anatomical Viewer in a variety of ways. What we're going to search for is one that I find easiest to think about now, is looking for specific genes within the database. And now I'm going to switch to just a new protein for fun. This one's called cadherin. So I typed in under gene keyword cadherin and clicked on Go. It searched the CGAP database and it found a bunch of different types of cadherin records. So let's look at the top one. The gene name is CDH1. That stands for cadherin 1 type 1 E-cadherin. So I'm going to click on the CDH1 link, and that will take me to the page where you can get to all the different types of information about this gene. The best tag that was identified for this gene is shown there on the left. So the nucleotides TG, T, G, 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 TG, CT are specific and unique for cadherin. I'm going to show you a picture in a minute so you can visualize this better, but that very short stretch of nucleotides is a tag that identifies cadherin 1. And so it's used to screen the database and to screen these microarrays and to develop this data information. The first one that we're going to look at, I'll show you, see the arrow there. If you click on LT Viewer, or the LTV, that stands for Ludwig Transcript Viewer. This shows you the RNA transcript from the cadherin gene, and there are 4 specific sequence tags that have been developed that are unique and can identify cadherin. The tag information is given below. So the one that we saw in the last one was tag number 1, TGT, GGG, TGCT. So that's tag number 1. And then it shows you the frequency that they found on the tag. So this one is at a frequency of 6488. Sometimes a tag can match to different genes, and they do warn you that the tag frequency can indicate that it's binding to more than one gene. But this data is confirmed later. What they're trying to do is find these short tags that are unique to every single individual gene. And so the tag that we're going to follow now is tag number 1. That's located there at the 3' end of the cadherin messenger RNA. So back to our table. We saw the LT viewer. Now let's look at what's called a digital northern to look at the RNA. So the northern is an experimental protocol where you extract RNA from a tissue, you run it on a gel, and then you use a probe to see if the RNA is present or not for a specific gene. It can tell you how good the expression is, whether there's a lot or little or no RNA. So now this is a in silico northern analysis of all the different tissues using that tag. The different colors indicate how many tags were present per 200,000. So what it's trying to do is give you, per 200,000 different messenger RNA molecules, how many could you expect to be your particular gene, in this case cadherin. And then it's searched all the different libraries that are present in the database, and I'm only showing you a few here, and it's giving you an idea of the level of expression. And so the pink color here means that it's up to or less than 256, and so high expressors would be greater than 512. So this is a pretty good expression, or pretty relatively high expressor if it's pink. So the top 4 samples, the first one being breast carcinoma epithelium, the second one being normal colon tissue, the third being also normal colon tissue, are expressing this at a relatively high level. And if you continue to look down in other samples, the expression of this particular gene is less. So this is a nice way to search for your gene of interest, to determine in these different samples if the gene is being expressed at a high level or at a low level. You can look, I clicked on the very top one, and you can look at the actual sample ID. And so this is the SAGE breast carcinoma epithelium, the very top one. It was expressing the cadherin at a relatively high level. It has 73,409 total tags in the library, and 30,000 are unique. That's pretty good. What that indicates is that they think there are about 30,000 different genes being expressed within this tissue, which is pretty high level. Back to the digital northern results, I've scanned down from the top just to show you that you get down to certain samples where it's being expressed at a low level. So we were up in the hundreds before, 200, 250 or so. And the column second from the right those numbers 36, 35, etc. Are the expression levels, and you can see we've gotten into the blue, and blue means very low expression. So that's the digital northern. That's pretty useful to determine what tissue is making what gene and at what level. Another way to look at this in an interesting way is the SAGE Anatomic Viewer. And you see the arrow there, you can click on the person, you can click on the cells, or click on both together. And this presents the data, I think, in sort of a neat way. I like visualizing things, make it easier to understand. And here they've organized the tissue into normal tissue or cancerous tissue. And all this data is present in detail. It's actually the records we've just been looking at. But now the data has been collated together so that we can visualize it. Again, it's expressing it in a number of tags per 200,000. So this cadherin is expressed at a really low level in the brain, in the retina. It's expressed at a higher level in thyroid from normal people, and at a lower level in the thyroid for people that have cancer. Looks like it's upregulated in the lung, in breast tissue, and in the pancreas and the kidney, and the cancer tissue compared to normal tissue. The stomach, it's expressed at a higher level in both. So sort of an interesting way, I think, to look at it. If you're studying breast cancer or pancreatic cancer, this information would be useful to you to look at genes that are either turned on or turned off in your particular tissue. Okay, now back to the digital northern results here. This is our tag that we've been following, the TGT, GGG, TGCT. I wanted to look at it in the lung, so I clicked on that, just to, the lung tissue, or you can look at it in, that's on the top. You can look at it in stomach tissue. For example, if you click on the different ones, it'll take you to each different category, and so you can make comparisons between the different tissues. So let's say you've gotten pretty interested now and you want to look in more detail. So rather than use the anatomic viewer, you want to look at gene expression in detail. So you're going to move down to the next database, which is the SAGE Digital Gene Expression Displayer, or DGED. Click on that link. That will take you to this page here. It's going to ask you these questions, and in this case you're going to compare two different samples to each other. I'm going to compare two SAGE library pools. If you have your own data in a very specific format, you can compare your data against the SAGE libraries, and that's where this probably becomes most useful to those that are doing this for a daily living kind of thing. Okay, here's what happens when you go into it. Select short tags. The long tag sometimes gives confusing information back and you get multiple genes, but just click on short tags. I'm going to list the libraries by tissue type, and you're going to compare pool A to pool B. So on the left, I'm choosing mammary gland or breast tissue from normal tissue, and that's pool A. And in pool B, I'm going to include the same tissue but from cancer. I'm going to exclude the cell lines because I just want to look at primary tissue taken from patients. And then click on Submit Query. Okay, it's going to give you a bunch of parameters here. We're going to accept the what's given to us. Use by changing the F factor and the Q factor, you can make it more or less stringent. So if you don't get very good results back, you can change these numbers to make it less strict. Or less significant, but if you keep it at the basis, you'll find something good. You can also search specific chromosomes. In this case, I'm going to search all chromosomes. This can take a little while, so you put your email address in here and it will send you a link by email. And then down to the bottom, click on Submit Query. Here, then, it's going to show you the different databases in the different pools that you can compare. And so if you remember, in pool A I wanted to compare normal tissue, and in pool B the cancer tissue. So what you want to do here is try to select matched tissues so that you're making a comparison between the same types of tissues. Then here are the results. So it tells you how many tags it searched in pool A and in pool B. And it gives you the tags themselves on the left hand side, the gene that the tag is from, the library that it was found in, and the sort of the expression levels of the tags themselves. If, oh, sorry, I didn't, at the top where it says get gene list, if you want to find, so this is showing all of them, but if you want to find those, for example, where it's higher in the cancer tissue, that was my pool B, than in the normal tissue, you click on the link here where B is greater than A, and this will show you all the tags that are expressed in the cancer tissue at a higher level than in the normal tissue. You can get from this the gene list of all the different genes in the sample and link out to the gene information in the CGAP database. And so you can find those that are expressed at a higher level, and then you can say, well, I don't know what alanine tRNA synthetase domain containing one protein is. Well, you need to go learn about it, and that's how you're going to do it, by looking at that. And linking back into the CGAP database and learning more about the protein in the gene itself. You can look at pathways, you can look at a variety of the data in a variety of different ways. So in this case, from this main result page, I'm going to scroll down, and there's, look at this row, cell motility. Well, what I was looking for is to find one that had a pathway associated with it. And so the first one I found there was the gene symbol is ARHGEF11. That's guanine nucleotide exchange factor rho. Click on that, and that'll show you the pathway. And there's rho right in the middle, and it's involved in exchanging guanine nucleotides onto GTP binding proteins, and involved in focal adhesion formation, stress fiber, cell movement. And so you can learn quite a bit here by following these pathways about your protein. Now we've linked back in at this point into the protein database, so you would then go back to what we talked about several weeks ago and learn more about the proteins. Okay, I'm going to end the lecture here. That there's so much stuff, but we focus it here on the Geo database and the Sage Genie database to look at particular genes. Your homework is here, and there's so much stuff. I want you to poke around in the databases a bunch, but I'm not going to make you write a huge report because it could be massive. All I want you to do is identify a gene that's differentially expressed between any two samples using the Geo database at NCBI. Tell me in which samples the gene is expressed at high levels or at intermediate levels, or is it not detected. Then use the Sage Genie to determine what tissues the gene is expressed in humans, and tell me what tag was used to identify the gene product. That's it. So hopefully it will be quick. As a last note, this won't be due until next Monday, so you have a week to do it.